Okay, so now we will be discussing the details of this particular application. Now listen. This is a fluidic system. This is what? This is sheath fluid, right? This is sheath fluid. This is what? Now listen. This is laser system. Okay. Now you should understand one thing. Suppose these are cell columns, single cell passing through laser one at a time, right? Now this is the laser. Now listen. This is the detecting system. This is the detecting system, forward scatter channel, and this is the detecting system known as side scatter channel. Okay, now listen. This forward scatter channel and this side scatter channel are attached to a computer screen. Attached to a computer screen. Now this is the axis of this screen. Okay, now you should understand one thing, that is, in the x-axis we will plot all those points which are associated with Texas red. That means in the x-axis, whenever any cell, suppose this is your CD8, with blue I will draw CD8, and with red suppose I will draw CD8, and with green suppose I will draw CD4. This is CD4 and this is CD8. Now listen. This is CD8 and CD4, and now to them antibodies are attached, and CD4 antibodies are attached to what? FITC. Attached to FITC, right? And CD8 antibodies are attached to Texas red. Now listen. Now listen. We now know that for your convenience, what I am doing is, I am actually not showing the CD protein, I am only showing the antibodies, and those green antibodies means those are CD4 antibodies. Okay, that means they are coupled to FITC, and red antibodies means they are coupled to Texas red, right? Suppose this is, now listen, suppose this is your this is your sample going down. Now listen, this is your sample which is going down. Now listen with all your attention. Suppose for when this particular cell was present here, when it was interacting with laser, what will happen is we will get a red signal, right? And this red signaling is coming because of Texas red. That means we will get a plot in this x-axis region, extreme x-axis that is here. This is the first. Now again we will get another plot here, but next time we are getting a green signal, right? That means now in the y-axis it is FITC tagged antibodies. Okay. Now what we will get is in the green, when green antibodies interact with laser, they will give out green fluorescence. That means here we will get one plot. Each plot corresponds to each cell. We have got how many cells? 1, 2, 3. First this one, second this one, third this one, right? And then it is again what? It is coming down. That means this cell has already interacted with what? Laser. That means again where we will get this or this. Now what is this? 1, 2, 3, 4, fifth cell. Fifth cell is coming. It is now coming down. Now where it will be placed? Sixth cell, where it will be placed? Okay. This is called scatter plot. We have already discussed yesterday. This is called scattered plot. Now listen with all your attention. Suppose in this line we have not got any double positive or double negative cell, right? Suppose I am omitting this line and I am putting another new, fresh, line of cell. I am suppose putting a fresh line of cell. Now let's talk about the first cell. Where the first cell will be produced? Where the first cell will give its scatter plot? In this area. Second cell in this area. Now where third cell? We know this, in this area. Now listen. This is the point you should understand, where you will get the scatter plot of this type of cells which are double positive. Where? In this area, right? Now we are drawing like this, okay? Half circle type and half circle type, okay? This is not the exact way to prepare the scatter plot, but for your understanding I am putting this. What you will get is in the computer screen some blue dots or black dots. That's it. Okay. Okay. This is the fifth one. Again, like this. Okay. Let us make it like this. Star. Again a star. It has got two antennas. That is a star. Okay. Now fifth one, sixth one. Again a star. Right. Again blue, star, right? Star, red, right? Clear? Now listen with all your attention. Now I will again remove all these cells. That means this has all gone down and we have actually taken their readings. I will again remove and I will again freshly put and I will again freshly analyze cells for the first cell. What will be this? A star. Second cell, star. Third cell, where? Just here. Double negative cells will be placed here. Why? Because FITC, the computer is not getting FITC signal or Texas rate signal. Okay. Now, fourth cell again this, fifth cell again here, sixth cell also here. This is so. Why I am saying all these things? Why it is important for you to understand these scatter plots? Now listen. Suppose a person has got leukemia, okay, and that person, in that person's flow cytometry analysis, we have got is more number of cells here. That means that person is having problem with T helper type 2 cells. That person is having problem with T helper type 2 cells. This occurs in what? Autoimmune disorders. That means that person is having autoimmune disease. Okay, this is the normal normal case where you will get double negative cells, in very small numbers, double positive cells in the okay numbers, because from double negative cells we know either CD4 or CD8 positive cells will arrive, right? That means they are nothing but what? Precursor of these, either of the two cells. But if we get less number of double positive cells, more number of particular Th2 cell, that means that particular cell is proliferating at a higher rate. That means the immune system is telling that cell that the immune system is always telling that cell to proliferate more. That means that particular cell has lost the control over its proliferation. There must be some problem with cell cycle. Okay. Now, in this context, I must say one thing, that is many people want to learn that how flow cytometry can help us in actually reading the number of cells undergoing cell division, or how flow cytometry can help us in understanding cell division. We will talk about this when we will be talking about cell division molecular mechanism in details, because right now this is very important for you to understand the cell types and all this with the help of flow cytometers. But in another video, when we will talking about cell division, you will learn that how using flow cytometry we can determine which cell is undergoing cell division, which cell is not undergoing cell division. There are special dyes called bromodiuridine. These dyes is very important. These are actually not dyes; these are actually chelating agents. They are actually, they will actually sit instead of thymine. Okay. Now we will talk about these things in details when we will talking about cell division. Right now, we know how these scatter plots are drawn when we want to specify cell types. Now listen with all your attention. If we do, now listen, if we change the axis, the problem is the whole scatter plot will be inverted, right? Now listen with all your attention. If we change the axis also, what will remain same? Negative and double positive cells. Their position will always remain same. That's a very basic thing you need to understand. Okay. So we will be now talking about another type of important application of flow cytometry. Okay.